Welcome back, everyone. I remain Professor Dixon Achimugumusa. In our last module, we took a comprehensive look at lipids, a chemically diverse group of molecules defined by their solubility in nonpolar solvents rather than by a shared structural backbone. We discussed their chemical properties, characteristics, reactions, and their biological roles. We also linked lipid chemistry to pathophysiology. You can find the link to Module 3 in the description below. Today we begin Module 4 and our focus shifts to a new class of biomolecules that are arguably the most functionally diverse in all of biochemistry: amino acids and proteins. Proteins are the molecular workhorses of the cell. Proteins act as enzymes, scaffolds, transporters, receptors, hormones, and antibodies. To understand proteins, we must first understand their building blocks, amino acids. Always keep in mind our guiding question: how does chemical structure determine biological function? By the end of this module, you should be able to recognize the structure and stereochemistry of the 20 standard amino acids, classify amino acid by polarity, charge, R group, chemistry, and nutritional essentiality. Explain properties such as deuterions, isoelectric point, and acid-base behavior. Describe peptide bond formation and its implication for protein structure. Understand the protein structure at 4 levels, from primary to quaternary structure. Distinguish fibrous, globular, and membrane proteins. And finally, outline the basic methods of protein purification and quantification. We begin our study of proteins at the most fundamental level, the amino acid. This is the general structure of the amino acid. Proteins are built from 20 standard amino acids, known as proteogenic amino acids. They all share common structure built on the alpha carbon, which is a tetrahedral carbon atom attached to 4 groups: an amino group, a carboxyl group, a hydrogen atom, and a variable side chain called the R group. The R group is what makes each amino acid unique, giving it distinct chemical properties and biological rules. Although many amino acids exist in nature, only these 20 are directly encoded by the genetic code and incorporated in proteins. Because the alpha carbon usually carries 4 different substituents, most amino acids are chiral. Glycine, however, is the exception, since its R group is just another hydrogen atom. In the Fischer projection, the alpha carbon is placed at the center, with the carboxyl group at the top of the R and the R group at the bottom. If the amino group projects to the left, the amino acid is designated L, as we can see in this diagram. If it projects to the right, it is designated D. Remarkably, nearly all amino acids in proteins are L configured, a universal feature of life known as homochirality. The amino acids do occur in nature, for example in bacterial cell walls and certain antibiotics, but they are not used in ribosomal protein synthesis. Chirality also gives amino acids the property of optical activity. When exposed to plain polarized light, a solution of chiral amino acid rotates the light either clockwise or counterclockwise. Understanding stereochemistry matters because it influences protein folding, enzyme specificity, and drug design. Now that we understand the general structure of amino acids, we can classify them based on the chemistry of their side chains. All 20 proteogenic amino acids share the same backbone, but their R group differs. And these differences determine polarity, charge, size, hydrophobicity, and ultimately their role in proteins. At physiological pH, amino acids are commonly grouped into five categories. Here we will focus on the nonpolar, aliphatic, and the aromatic groups. The nonpolar aliphatic amino acids have side chains made mostly of carbon, hydrogen, making them hydrophobic. They cluster inside proteins away from water and help stabilize folded structures. Glycine, the simplest amino acid, adds flexibility because its side chain is just a hydrogen atom. Alanine, with a small methyl group, is abundant and fits easily into tight spaces. Valine, leucine, and isoleucine have branched side chains that are strongly hydrophobic. Isoleucine is notable for having two chiral centers. Proline is unique. Its side chain loops back to bond with its own amino group, creating a rigid ring that introduces a bend in protein chains. Methionine, though it contains sulfur, is still considered nonpolar and plays a special role as the first amino acid added during protein synthesis. The aromatic amino acids, that is the phenylalanine, tyrosine, and tryptophan, carry large ring structures in their side chains. These rings are mostly hydrophobic but can also participate in special interactions. Phenylalanine is purely nonpolar and often buried in protein cores. Tyrosine resembles phenylalanine but has a hydroxy group that makes it weakly polar and a target for phosphorylation in signaling pathways. Tryptophan has the largest side chain, an indole ring that is mostly hydrophobic but can form hydrogen bonds. Though rare in proteins, tryptophan is biologically important as a precursor of serotonin and niacin. We continue with our classification of the 20 standard amino acids by the chemistry of their side chains. Some are polar but uncharged at physiological pH. Their side chains contain electronegative atoms, such as oxygen or nitrogen, which allow hydrogen bonding without carrying a net charge. Because they are hydrophilic, these amino acids are often found on protein surfaces or in enzyme active sites. Serine and threonine both carry hydroxy groups and are common sites of phosphorylation. Threonine is also notable for having two chiral centers. Cysteine contains a thiol group that can form covalent disulfide bonds, stabilizing protein structures. Asparagine and glutamine, derived from the acidic amino acids, carry imide groups that are polar but uncharged, with asparagine serving as a key site for glycosylation. The acidic amino acids, aspartate and glutamate, have side chains with carboxyl groups that are deprotonated at physiological pH, giving them a negative charge. These residues form ionic interactions, stabilize protein structures, and play roles in metabolism and neurotransmission. The basic amino acids, lysine, arginine, and histidine, carry positively charged side chains. Lysine and arginine are strongly basic and frequently interact with DNA due to their positive charges. Histidine is chemically versatile because its imidazole group has a pKa near physiological pH, allowing it to act both as a proton donor and acceptor in enzyme catalysis. Beyond chemistry, amino acids are also classified nutritionally. Non-essential amino acids are synthesized by the body, while essential amino acids must be obtained from the diet. The nine essential amino acids include histidine, leucine, isoleucine, lysine, methionine, phenylalanine, threonine, and tryptophan, and valine. Some amino acids are conditionally essential, meaning they become necessary during growth, illness, or stress. For example, cysteine depends on methionine availability, and tyrosine depends on phenylalanine. Arginine is also conditionally essential, especially in children and during catabolic stress. Deficiency in essential amino acids can impair protein synthesis, growth, and immune function, highlighting their importance in nutrition and health. Now that we have classified amino acids by their side chains, the key questions becomes, why does this matter biologically? The answer is that the chemical nature of each chain shapes how proteins fold, stabilize their structures, catalyze reactions, and interact with other molecules. Hydrophobic side chains, such as those of valine, leucine, isoleucine, phenylalanine, and methionine, drive protein folding through hydrophobic effect. These residues clusters inside proteins, forming a stable core, shielded from water. If this core is disrupted, proteins often unfold and lose function in a process called denaturation. Proline, with its rigid ring, introduces bend and turns, helping proteins change direction in their chains. Polar side chains add hydrogen bonding capacity. Serine, threonine, asparagine, and glutamine stabilize structure and active sites, while tyrosine and tryptophan bridge hydrophobic interiors with aqueous surfaces, thanks to their dual hydrophobic and hydrogen bonding nature. Charged side chains like those of aspartate, glutamate, lysine, arginine, and histidine form ionic bonds or salt bridges that stabilizes higher order structures and mediate interaction with DNA, RNA, and other proteins. Histones rich in lysine and arginine bind tightly to DNA, a key feature of gene regulation. Aromatic residues like phenylalanine, tyrosine, tryptophan absorb ultraviolet light, a property exploited in measuring protein concentration. Tryptophan also fluoresces, allowing researchers to probe protein structure and interactions. The diversity of amino acid side chain is the foundation of protein function. Every fold, every catalytic event, every interaction a protein makes can be traced back to the chemistry of the amino acids. Beyond their structures, amino acids have physical and chemical properties that determine how they behave in solution, how they can be separated in the lab, and how they influence protein stability in cells. All 20 standard amino acids are crystalline solids with high melting points. In both solid and aqueous form, they exist mainly as dipolar ions rather than neutral molecules, which explain their solubility in water, but poor solubility in nonpolar solvents. Some amino acids even contribute to taste. Glycine and alanine are sweet, while glutamate gives the savory flavor known as umami. Aromatic amino acids absorb ultraviolet light, a property widely used to measure protein concentration. Chemically, amino acids are amphoteric, meaning they can act as both acids and bases. Their carboxyl group donate protons, while the amino group accept them, allowing amino acid to buffer changes in pH. At low pH, both groups are protonated and the molecule carries a positive charge. As pH rises, the carboxyl group loses its proton, producing a zwitterion with both positive and negative charges, but no net charge. At high pH, the amino group also loses its proton, leaving the molecule negatively charged. The isoelectric point, Pi, is the pH at which an amino acid has no net charge and exists mainly as a zwitterion. Below the Pi, it is positively charged and moves towards the cathode in an electric field. Above the Pi, it is negatively charged and moves towards the anode. This principle informs techniques such as isoelectric focusing, which separates protein based on their Pi values. Titration curve reveals the pKa values of amino acids. Simple amino acids like alanine show two inflection points corresponding to the carboxyl and amino groups with a Pi line between them. Amino acids with ionizable side chains such as histidine or lysine display three inflection points reflecting the additional group. Amino acids contribute to buffering capacity in biological systems. A molecule buffers best near its pKa, where protonated and deprotonated forms are balanced. Histidine is especially important because its side chain has a pKa close to a physiological pH, making it an effective buffer in proteins such as hemoglobin, which helps maintain blood pH during carbon dioxide transport. Other side chains include those of glutamate, aspartate, lysine, and arginine also play roles in protein buffering. Amino acids behave in a fascinating way depending on pH. And the key to understanding this is the concept of the zwitterion. A zwitterion is a molecule that carries both a positive and a negative charge at the same time, yet has no overall net charge. In water, amino acid rarely exist in a neutral form. Instead, the carboxyl group donates a proton to the amino groups, producing COO- and NH3 plus. This internal charge separation is more stable than the neutral form and explains why amino acids have high melting points and dissolve well in water. Glycine, The simplest amino acid illustrates this behavior. At very low pH, glycine is fully protonated and carries a positive charge. As the pH rises to about 2.3, the carboxyl group loses its proton and glycine shifts into its zwitterionic form. At its isoelectric point around 5.97 pH, glycine exists almost entirely as a zwitterion and has no net charge, so it does not move in an electric field. Raising the pH further to about 9.6 causes the amino group to lose its proton, giving glycine a net negative charge. This stepwise ionization is universal to amino acids, though those with ionizable side chains show an additional transition that shifts their isoelectric point toward acidic or basic. The pKa value of each ionizable group tells us that the pH at which half of those groups are deprotonated. Carboxyl groups typically have a low pKa values around 2, meaning they release proton easily. Amino groups have higher pKa values around 9 to 10, meaning they hold on to protons until the solution is more basic. Side chains vary widely. Aspartate and glutamate have acidic pKa values near 4. Histidine imidazole group is near 6. Cystane's thiol is around 8, and so on. It is important to remember that these values can shift when amino acids are parts of protein. By knowing pKa values, we can predict the charge states of amino acids at any pH, which in turn helps us understand the behavior in separation techniques, their interaction with other molecules, and their contributions to protein function. The reactivity of amino acid is central to both biology and laboratory science. Their chemical groups undergo characteristic reactions that drive processes such as neurotransmitter synthesis, protein modification, and analytical detection. The alpha carboxyl group behaves like other carboxylic acids. It can form esters with alcohols, a reaction used in preparing amino acids and derivatives for analysis. In cells, esterification links amino acid to tRNA, that is the transfer RNA, a crucial step in protein synthesis. Most importantly, the carboxyl group reacts with the amino group of another amino acid to form a peptide bond. The peptide bond is the fundamental linkage in proteins. It can also undergo decarboxylation, releasing carbon dioxide, a reaction that produces biologically active amines. The alpha amino group also shows distinctive chemistry. It can be acylated to form amides, and it reacts with anhydrine to produce purple color, a classic test for amino acid. Side chains add further diversity. Cystane's thiol group form disulfide bonds, stabilizing proteins. Tryptophan's indole ring can be selectively modified to probe its role in proteins. Two transformations are especially important in metabolism. Decarboxylation converts amino acid into biogenic amines, many of which are neurotransmitters. Histidine to histamine, glutamate to GABA, tyrosine to dopamine, and adrenaline. Transamination transfers amino groups between molecules, linking amino acid metabolism to carbohydrate metabolism. Enzymes such as ALT and AST catalyze this reaction, and the activity in blood is clinically to assess liver function. Several diagnostic tests exploit amino acids chemistry, while modern techniques rely on chromatography and mass spectrometry. The reactions of amino acids are the molecular foundations of life. When these reactions go wrong, the consequences can be serious, even life-threatening. Let's look at four examples that show how amino acid chemistry connects directly to biology and medicine. Decarboxylation. Decarboxylation reaction produces key neurotransmitters such as dopamine, serotonin, and GABA. When these reactions fail due to genetic mutations, vitamin B6 deficiency, or enzyme inhibition, neurotransmitter levels drop, leading to neurological disorders. For instance, aromatic L-amino acid decarboxylase deficiency prevents the formation of dopamine and serotonin, causing severe movement and developmental problems. Impaired glutamate decarboxylation reduces GABA, leading to seizures, as seen in pyridoxine-dependent epilepsy. Clinically, drugs like carbidopa exploit this chemistry by blocking dopamine formation outside the brain, ensuring more L-dopa reaches the brain in Parkinson's therapy. Transamination defects. Transamination reactions transfer amino groups, linking amino acid metabolism to energy pathways. Defects in this reaction can disrupt nitrogen disposal and energy balance. Clinically, elevated ALT/AST levels in blood are used as markers of liver damage, since these enzymes leak out when hepatocytes are injured. Broader defects in transamination contribute to metabolic diseases that often appear in infancy. Cysteine oxidation and protein misfolding. Cysteine's thiol group normally stabilizes proteins through disulfide bonds, but under oxidative stress, excessive or incorrect disulfide formation leads to misfolding and aggregation. This chemistry underlies cataracts, where crystalline proteins in the lens aggregate, and neurodegenerative diseases like Parkinson's, where oxidative damage promotes toxic protein inclusions. The endoplasmic reticulum normally controls disulfide formation via protein disulfide isomerase, but when overwhelmed, the endoplasmic reticulum stress triggers cell death, contributing to diabetes, neurodegeneration, and cancer. Antioxidants such as glutathione exist precisely to protect cysteine and other sensitive residues from uncontrolled oxidation. Phosphorylation and cancer pathways. Phosphorylation of serine, threonine, and tyrosine residues is the cell's main way of regulating signaling pathways. Kinases add phosphate group, phosphatases remove them, and together they control growth, survival, and metabolism. When phosphorylation is dysregulated, cancer can result. Having explored amino acids in detail, we now turn to how they join together to form peptides and ultimately proteins. This step is crucial because it is through peptide bonds that the chemical diversity of amino acid is translated into the functional diversity of proteins. What are peptides? Peptide is formed when two or more amino acids are covalently linked. Small chains are called dipeptides, tripeptides, or oligopeptides, while longer chains are called polypeptides. Proteins are essentially long polypeptides that fold into specific three-dimensional structures, giving them unique biological functions. Once amino acids are incorporated into a chain, they are referred to as residues, since part of their structure is lost during bond formation. The peptide bond is an amide linkage between the carboxyl group of one amino acid and the amino group of another, with the release of water. This condensation reaction is energetically unfavorable in water. So in living cells, it is powered by the ribosome. Amino acids are first activated as amino acyl tRNAs, and the energy stored in this linkage drives peptide bond formation. The ribosome's peptidyl transferase activity ensures precise positioning and catalysis, extending the growing chain one residue at a time. The peptide bond have two distinct ends. The N terminus with a free amino group and the C terminus with free carboxyl group. By convention, sequences are always written from the N to C. During protein synthesis, chains also grow in this direction. This directionality is fundamental. A dipeptide written as ALAGLY is chemically and functionally different from GLYALA. In larger proteins, the exact sequence of residues read from N to C uniquely determines the protein's identity, structure, and function. Now that we know peptide bonds form, how they are formed, let's look more closely at their structure. The peptide bond is not just a simple link between amino acids. Its electronic properties impose strict geometric rules that make protein structures possible. Although the peptide bonds look like a single CN bond next to a carbonyl group, resonance changes the picture. The lone pair of electrons on nitrogen delocalizes into the carbonyl group, giving the CN bond partial double bond character. This makes the bond shorter and stronger than a typical single bond and prevents free rotation. In effect, the peptide bond behaves like a rigid double bond, locking atoms in place. Like a true double bond, the peptide bond can adopt cis or trans configuration. Almost all peptides are trans because Cis brings adjacent side chains too closely together, creating steric strain. The exception is bonds between proline, where the difference between cis and trans is smaller. About 5 to 10% of proline containing peptide bonds are cis, introducing bends that are important for protein structure. The peptide bond is remarkably stable. Under normal physiological conditions, it resists hydrolysis for hundreds of years, ensuring that proteins maintain their integrity once formed. Hydrolysis does occur under extreme conditions, such as strong acid or base treatments, or through the action of proteases. These enzymes are essential in digestion, protein turnover, blood clotting, and immune defense, each with specific preferences for where they cut. Beyond stability, the peptide bond is polar, with its carbonyl oxygen and amide nitrogen able to form hydrogen bonds. This polarity is the foundation of secondary structures like alpha helices and beta sheets. Several small peptides illustrate how even short chains can have profound biological roles. Glutathione, for instance, is a tripeptide of glutamate, cysteine, and glycine, and is a major antioxidant. Its cysteine thiol group neutralizes reactive oxygen species, and two molecules can join to form oxidized glutathione, later recycled back to its reduced form. Glutathione also aids in detoxification and protects protein from oxidative damage. Deficiency leads to anemia, infection risk, and neurodegeneration. Oxytocin is a 9 residue peptide with a disulfide ring produced in the hypothalamus and released from the pituitary gland. It stimulates uterine contraction during labor and milk ejection during breastfeeding. Beyond reproduction, oxytocin influences social bonding and emotional responses. Though its roles are complex, clinically it's used to induce labor and prevent postpartum hemorrhage. Closely related is vasopressin, which is also a 9 residue peptide with a similar structure but distinct function. It regulates water balance by increasing reabsorption in the kidney and can raise blood pressure through vasoconstriction. Deficiency causes diabetes insipidus, marked by excessive urine output and thirst, while excess secretion leads to dangerous water retention and hyponatremia. Insulin demonstrates how peptides scale into small proteins. Composed of two chains linked by disulfide bonds, insulin regulates blood glucose by promoting uptake, storage in tissues. It synthesizes proinsulin, which is processed into active insulin and C-peptide. Deficiency or resistance leads to diabetes, one of the most significant global health challenges today. The discovery of insulin transformed type 1 diabetes from fatal to manageable, a landmark in medical history. We now move from peptides to proteins, which are the most complex and versatile macromolecules in biology. Everything we have studied about amino acids and peptides bond leads to this point because proteins are simply long chains of amino acids whose sequence and arrangement give rise to extraordinary structural functional diversity. Proteins are large molecules composed of one or more polypeptide chains. They vary enormously in size from small hormones like glucagon, which has about 50 residues, to titin, the largest known protein with over 34,000 residues in a single chain. Their molecular weight ranges from thousands to millions of Daltons. Each protein sequence is encoded by a gene. Proteins are built mainly from carbon, hydrogen, oxygen, and nitrogen, with sulfur present in cysteine and methionine. Some proteins also contain phosphorus or metal ions, such as iron, zinc, or magnesium, which acts as cofactors. Nitrogen content is relatively constant at about 16%, a property used in protein quantification. The human genome encodes roughly 20 to 25,000 protein coding genes, but alternative splicing and post-translational modification expand this into hundreds of thousands of distinct proteins. Proteins are the most abundant organic molecules in cells, making up about half of cellular dry mass. Proteins share several important characteristics. They are amphoteric, carrying both acidic and basic groups, and each has a specific isoelectric point where its net charge is zero. Because of their size, proteins form colloidal dispersions in water rather than through solutions, scattering light in the process. They can be denatured by heat, pH extremes, or chemicals, losing their structure and usually their function, though sometimes this process is reversible. Protein also give characteristic color reaction used in detection and quantification. We will now examine the major categories of protein functions, providing specific examples. Keep in mind that the function of every protein ultimately traces back to the precise three-dimensional shape that its amino acid sequence directs it to adopt. Enzymes are biological catalysts, accelerating reactions that would otherwise be far too slow to sustain. They achieve this by binding substrate to precisely shaped active sites, stabilizing transition states, and lowering activation energy. Their specificity is remarkable. Most enzymes act only on one reaction or a closely related set. Examples include hexokinase, which initiates glycolysis by phosphorylating glucose, DNA polymerase, which replicates genetic material with high fidelity, and digestive enzymes like pepsin and trypsins. Enzymes are the engines of metabolism. Structural proteins provide strength and resilience. Collagen is the most abundant protein in the body and forms rope-like fibrils that resist stretching and give tensile strength to connective tissues. Elastin provides elasticity, allowing tissues like skin and blood vessels to stretch and recoil. Keratin forms a tough outer layer of skin, hair, and nails. Actin and tubulin build a cytoskeleton, maintaining cell shape and enabling movement. Defects in structural proteins lead to diseases such as scurvy, Ehlers-Danlos syndrome, and brittle bone diseases. Proteins also serve as carriers. Hemoglobin transports oxygen in the blood with cooperative binding that makes oxygen delivery highly efficient. Serum albumin carries fatty acids, hormones, and drugs. Transferrin delivers iron, while lipoproteins transport cholesterol and other lipids. Within cells, myoglobin stores oxygen in muscle and membrane transporters like GLUT4 and sodium potassium ATPase regulate nutrients ion movement across membranes, maintaining homeostasis. Motor proteins convert chemical energy into mechanical work. Myosin interacts with actin filaments to drive muscle contraction powered by ATP hydrolysis. Kinesin and dynein move cargo along microtubules, organizing the cell's interior and enabling axonal transport in neurons. Disruptions of this system contribute to neurodegenerative disease. Antibodies or immunoglobulins recognizes and neutralize foreign antigens with exquisite specificity. Complement protein form cascades that destroy pathogen while cytokines and interferons coordinate immune responses. These proteins are the molecular defenders of the body. Many hormones are proteins or peptides. Insulin and glucagon regulate blood glucose. Growth hormones drive development. Erythropoietin stimulates red blood cell production. And pyrothyroid hormone controls calcium balance. Leptin signals nutritional status to the brain. Each hormone acts by binding to a specific receptor, ensuring precise communication between cells. Some proteins store essential molecules for later use. Ferritin safely stores iron. Casein in milk provides nutrients for infants. Ovalbumin in egg white nourishes embryos. And seed storage proteins like glutenins provide reserve for germinating plants. Proteins are not only catalyst carriers or structural components, they are also central players in signaling and regulation. Because proteins are so diverse, biochemists classify them using three complementary frameworks: by shape, by chemical composition, and by biological function. Together, this system provides a complete picture of protein's identity and role. Proteins fall broadly into fibrous, globular, and membrane categories based on shape. Fibrous proteins are elongated, highly ordered, forming strong or elastic structures. Collagen with its triple helix, rich in glycine and proline, provide tensile strength to connective tissues. Silk fibrin built from small amino acids like glycine and alanine packs into beta sheets that yield strength and flexibility. Globular proteins, in contrast, fold into compact spherical shapes. They are generally soluble with hydrophilic residues on the surface and hydrophobic residues buried inside. This architecture creates active site binding pockets and recognition surfaces, the basis for enzymes, antibodies, and transport proteins. Membrane proteins are embedded or attached to lipid bilayers. Their transmembrane regions are rich in hydrophobic residues, allowing them to span the nonpolar interior of membranes. They mediate transport, signaling energy conversion and adhesion, with structures adapted to the unique environment of the membrane. Proteins can also be divided into simple and conjugated types. Simple proteins yield only amino acids upon hydrolysis, such as albumin and globulins. Conjugated proteins contain non-protein protestic group essential for activity. Examples include glycoprotein with carbohydrate chains, lipoproteins that transport lipids, hemoproteins like hemoglobin and with iron containing heme, flavoprotein with flavin cofactors, and metalloproteins that coordinate metal ions for catalysis or structure. Proteins are grouped by their roles they perform into enzymes, structural proteins, transporters, motors, storage proteins, receptors, signaling molecules, and immune defense system. These categories highlight the breadth of protein activity across biology. Protein function depends entirely on three-dimensional shape, that sh- and that shape is detected by sequence. To make sense of this relationship, biochemists describe protein structure at four levels: primary, secondary, tertiary, and quaternary. Here we'll focus on the first two. The primary structure is simply the linear sequence of amino acids in a polypeptide chain, read from the N terminus to the C terminus. The sequence is encoded by DNA and established during ribosomal synthesis. Peptide bonds, strong covalent linkages, hold a chain together, making primary structure highly stable under physiological conditions. Even a single substitution can have dramatic effects. Sickle cell anemia is a classic example. Replacing glutamate with valine at position 6 of hemoglobin's beta chain creates a hydrophobic patch that drives abnormal polymerization, distorting red blood cells and causing disease. This illustrates how changes at primary level can ripple through all higher structures. Primary structure is determined experimentally, though gene sequencing, mass spectrometry, or historically by Edman degradation. Secondary structure refers to local repetitive folding patterns stabilized by hydrogen bonds between backbone, amide, and carbonyl groups. Side chains project outward and do not define these structures directly. The two principal types of secondary structure are the alpha helix and the beta sheet. The alpha helix is a right-handed coil stabilized by hydrogen bonds between carbonyl oxygen of one residue and the amide hydrogen of four residues above. Alpha helices are abundant in fibrous protein like keratin and in globular proteins such as hemoglobin. The beta sheet consists of extended strand linked by hydrogen bonds between adjacent backbones. Beta sheets are found in silk, fibrin, membrane porins, antibody domains, and enzymes active sites. Other secondary structures include betatons. These often form binding sites and play critical roles in protein function. The central question in biochemistry is how a newly synthesized polypeptide chain emerging from the ribosome as a linear string of amino acids finds its correct three-dimensional shape, and what happens when the process fails. The answer lies in thermodynamics, kinetics, and cellular quality control systems. Proteins fold spontaneously into native conformation because of the state has the lowest Gibbs free energy. Folding is stabilized by favorable enthalpic interactions, hydrogen bonds, ionic contacts, van der Waals forces, and by the hydrophobic effect which drives nonpolar side chains into the protein interior. Although folding reduces the entropy of the chain itself, the release of ordered water molecules around hydrophobic residues increases solvent entropy, making folding energetically favorable. Proteins do not fold in a random fashion or by trial and error. Instead, folding follows defined pathways along an energy landscape, like a funnel, guiding chains towards the native state. Early events include formation of local secondary structures and hydrophobic challenges. Although sequences contains all the information for folding, the crowded cellular environment poses risk of aggregation. Molecular chaperones prevent misfolding by shielding exposed hydrophobic regions and providing protected environments. Specialized chaperones in the endoplasmic reticulum assist secreted and membrane proteins. Chaperones are upregulated under stress. The heat shock, the heat shock response to protect cells when proteins are at risk of unfolding. Proteins depend on their precise three-dimensional structures for function. Denaturation is the unfolding of the secondary, tertiary, and quaternary structure without breaking the peptide bonds because the non-covalent forces stabilizing proteins are relatively weak. They are easily disrupted by changes in the environment. Denaturation destroys the native fold and usually abolishes biological activity. Denaturation agents act in different ways. Heat increases the molecular motion. breaking weak interaction at high concentration. Unfolded chains aggregate irreversibly as seen in cooked egg. Extreme pH alters iodization states, disrupting salt bridges and hydrogen bond. Cheotropic agents like urea and guanidinium chloride interfere with hydrogen bonding and the hydrophobic effect causing complete unfolding. Detergents such as SDS insert into hydrophobic cores, disrupting packing and coating proteins with negative charge, a principle used in SD page. Reducing agents like DTT break disulfide bonds, destabilizing proteins that rely on them. Organic solvents disrupt hydrophobic interaction and hydrogen bonding, often causing precipitation. The naturation can be reversible if conditions are mild and protein refolds once the agent is removed, demonstrating that sequence encodes the native fold. To study proteins or to produce them for therapeutic use, we must first isolate them from complex biological mixtures. Protein purification is therefore a cornerstone of biochemistry, combining principles of cell disruption, fractionation, and separation. The process begins with breaking open cells to release proteins. Methods include mechanical disruption by homogenization, sonication, freeze thaw, French press, etc. And we have chemical disruption with detergents or solvents and enzymatic digestion of cell walls. The resulting lysate contains thousands of proteins plus nucleic acids, lipids, and metabolites. Centrifugation removes insoluble debris, leaving a soluble extract for purification. Centrifugation separates cellular components by size and density. Differential centrifugation uses progressively higher speeds to pellet cell debris, organelles, and ribosomes. Density gradient centrifugation, often with sucrose, separates particles by buoyant density, while analytical ultracentrifugation provides precise measurements of protein mass and sedimentation. Precipitation exploits differences in solubility. Ammonium sulfate salting out is widely used. High salt concentration reduces protein-water interaction, causing proteins to aggregate and precipitate at characteristic salt level. Isoelectric precipitation adjusts pH to protein's isoelectric point, minimizing charge and solubility. Chromatography is the most versatile tool for purification. Ion exchange chromatography separates proteins by charge, binding them to charged resins and eluting with salt or pH changes. Gel filtration or size exclusion separates by size. Large proteins elute first, small ones later. Affinity chromatography achieves high specificity by immobilizing ligands that bind the target protein. Immobilized metal affinity chromatography is widely used to purify recombinant protein with histidine tags. Hydrophobic interaction chromatography separates surface hydrophobicity under high salt condition. Reverse phase HPLC provide high resolution separation of proteins and peptides. Electrophoresis is a powerful technique for separating protein based on how they move through a gel under the influence of an electric field. Protein quantification method, which we will examine in detail later, provide ways to measure protein concentration after separation. The detection, identification, and measurement of proteins and amino acids are fundamental tasks in biochemistry laboratories, clinical laboratories, and food science. A wide range of chemical tests has been developed for those purposes, exploiting the distinctive chemical properties of amino acid side chains, the peptide bonds, and other structural features of proteins. These tests can be broadly divided into qualitative tests, which establish the presence or absence of protein or specific amino acid, and quantitative tests, which measure the amount of proteins present in a sample. Qualitative test is very important in biochemistry, and we have quite a number of qualitative tests that we carry out in biochemistry. Biored test, a general detector for proteins and polypeptides. In strong-based copper ions, bind peptide bonds to give a violet color. Positive means two or more peptide bonds are present. Free amino acids usually don't give strong purple. More peptide bonds and higher protein concentration deepen the color. The ninhydrin test is the most general qualitative test for amino acids. Ninhydrin oxidizes amino acids and forms Romance purple with the released ammonia. Most alpha amino acids are purple except proline and hydroxyproline, which yield a yellow-brown color due to their ring structure. A purple color signals amino acids. Yellow-brown signals proline and hydroxyproline, also used in chromatography and older amino acid analysis. The xanthoproteic test detects aromatic amino acids such as tyrosine, tryptophan, phenylalanine by nitrating their rings with concentrated nitric acid, producing a yellow color that turns orange in alkaline solution. Millon's test is specific for tyrosine, yielding a white precipitate that becomes brick red upon heating due to formation of mercury complex. The Sakaguchi test identifies arginine through its guanidinium group, which reacts with naphthol and an oxidizing agent to produce a bright red color. The Sullivan test distinguishes cysteine, whose free thiol groups react with sodium nitroprusside to give a red or purple color. While cysteine remains negative, the lead acetate test provides a general assay for sulfate-containing amino acids, forming a black lead sulfide precipitate when proteins are hydrolyzed under alkaline conditions. The poly test detects histidine and tyrosine by coupling their rings with diazotized sulfonylic acid to yield a red azodine. Tryptophan can be identified by Hopkins cold test, which produces a purple ring at the interference of solution, while glycoxylic acid reacts with the indole ring in sulfuric acid, or by the Ellick test, which uses which is used to generate a purple color. Accurate measurement of protein concentration is one of the most routine but essential tasks in biochemistry. Several assays are available, each exploiting different chemical or physical properties of protein, offering distinct strengths and limitations. The simplest method is ultraviolet absorbance at 280 nanometers, which relies on the strong absorbance of aromatic residues such as tryptophan and tyrosine. It is rapid, non-destructive, and requires no reagents, but nucleic acids can interfere and proteins lacking aromatic residues may be underestimated. The biuret acid based on reaction of peptide bonds with copper ions in alkaline solution produces a purple complex measurable at 540 to 550 nanometer. Though relatively insensitive and requiring large amounts of protein, it is reproducible and widely used in clinical chemistry for serum protein determination. The Lowry assay improves sensitivity by combining the biuret reaction with reduction of the Folin-Siocalcio reagent, producing an intense blue color. It is far more sensitive than the biuret, but prone to interference from sugars, lipids, buffers, and detergents. The Bradford assay developed later uses Kumasi brilliant blue, which shifts from red to blue upon binding protein. It is fast, simple, and sensitive, but its response varies with protein's composition and can be affected by detergents. The BCA assays combine the biuret reaction with chelation of reduced copper by BCA, yielding a purple complex at 562 nanometer. It is highly sensitive, compatible, compatible with many detergents, and suitable for membrane proteins through reducing agents, sugars. Reducing agents and sugars can cause false positive. The Jedal method, which is a classical approach, determines protein content by measuring total nitrogen after acid digestion. It is highly accurate and remains the reference method in food and agricultural analysis, though it is labor intensive and cannot distinguish protein nitrogen from other sources. Enzyme-linked immunosorbent assays provide extraordinary sensitivity and specificity for quantifying individual proteins in complex mixtures, detecting concentration down to picogram per millimeter range. Unlike general assays, enzyme-linked immunosorbent assay is tailored to specific targets such as hormones, antibodies, or pathogen proteins, making it indispensable in clinical diagnostics. Full biochemical characterization of protein goes beyond simply measuring its concentration. It requires its molecular weight, analyzing its amino acid composition, and ultimately establishing its complete sequence. Together, these analyses define protein's primary structure and provide a foundation for understanding its higher order folding, function, and interaction. Molecular weight can be estimated by several experimental methods. Amino acid analysis determines the overall composition of a protein, not the sequence. Classical analysis involves complete hydrolysis of the protein, separation of the amino acid by chromatography, and detection with reagents such as denhydrin or modern fluorescent tags. Protein sequencing established the exact order of amino acid in a chain. Classical sequencing relied on Edman degradation, a stepwise chemical method that removes and identifies one residue at a time from the N-terminus. Because proteins are too long to sequence directly, they are first cleaved into overlapping fragments using enzymes or chemical reagents. Today, mass spectrometry has largely replaced Edman chemistry, with tandem mass spectrometry providing rapid sensitive sequencing of peptides and enabling large-scale proteomics. In the final stage, protein characterization, we will now move our attention to determining the three-dimensional structure of protein at atomic resolution and establishing whether a purified preparation is truly homogeneous and suitable for study. While knowing the amino acid sequence is essential, it does not reveal the actual spatial arrangement of atoms. For this, structural biology relies on three principal techniques: X-ray crystallography, nuclear magnetic resonance spectroscopy, and cryo-electron microscopy. Before protein can be used for biochemical characterization, structural studies, or therapeutic application, it must be shown to be pure, meaning homogeneous in identity, molecular weight, and conformation. Establishing purity requires multiple independent criteria, since no single test is fully definitive. And this completes Module 4 on amino acids and protein biochemistry. We have traced the journey from chemistry of amino acids and peptide bond through the hierarchy of protein structure, the principle of folding and misfolding, the full range of methods for isolation, purification, and sequencing and structural determination. This provides the foundation for modern protein science and its application in medicine and biotechnology. Deepen and consolidate your understanding of the material covered in this module. You are strongly encouraged to consult the following standard biochemistry textbooks. These texts provide expanded explanation, worked examples, clinical correlations, and practice questions that will reinforce and extend what you have learned. Please use the comment section to post your questions, observations, or any points from this module that require further clarification. Thank you for your attention. I look forward to seeing you in Module 5.